In our previous lesson, we looked at the structure of fatty acids and referred briefly to their efficiency in terms of energy storage. Now we want to look at the class of lipids that represent the actual storage form of fatty acids. Triacylglycerols, or triglycerides, are formed by esterifying three fatty acid chains to the glycerol backbone. As you can see from the structure on the far left, glycerol is a 3-carbon compound with 3 hydroxyl groups. Each fatty acid is attached to the triacylglycerol by means of an ester bond. Recall that lipids are amphipathic, with separate polar and nonpolar domains. In the case of triglycerides, the polar portion is comprised of the ester groups, and the tails are highly nonpolar. You can get a better idea of the size and geometry of a triglyceride by examining the space-filling models below. The primary storage place of triacylglycerols is in adipose tissue. To retrieve the fatty acids as a nutrient source, enzymes known as lipases must hydrolyze the ester linkages connecting them to the glycerol backbone. Fatty acids serve as concentrated stores of metabolic energy, primarily due to the highly reduced nature of their nonpolar tails. We will discuss this in greater detail when we consider lipid metabolism. In contrast to enzymatic cleavage, illustrated on the left in our figure, saponification is the process of chemically cleaving a triglyceride, as on the right. When a strong base like sodium hydroxide is applied to a triglyceride, the fatty acid chains are released as sodium salts. These are soaps, and when used with hard water, can precipitate calcium and magnesium ions. This is the scum left in sinks and bathtubs. The glycerol component of triacylglycerols is a common component in creams and lotions and is also used in the manufacture of nitroglycerin. We can see that triglycerides have both metabolic and chemical roles to play.